Good morning, everyone. Welcome to today's session on language, anatomy, networks, and pathology. My name's Greg de Zubicare, and I'm delighted to co-chair this session with my colleague, Nicola Palomero-Gallagher. This morning, all of the speakers that you'll be hearing will be speaking for 10 minutes, and there'll be 2 minutes for questions afterwards. And you can actually use the microphone up the back there in preference to the app. But we'll be keeping people strictly on time. Now, as is customary, I would like to pay our respects to the Indigenous elders, the Turrbal and Yagara people, on whose land we are now meeting. Our very first speaker is Loïc Labache. Yeah. This is Loïc's first time in Australia, so I hope he's enjoying it. Loïc's... I don't have your title, so if we can have the first slide. Yes, so Loïc's first talk today is on the molecular and cellular underpinnings of human brain lateralization. So please take it away.
Thank you. So good morning, everyone. So my name is Loïc Labache, and I am a postdoctoral researcher in the Department of Psychiatry at Rutgers University. And today I'm going to talk to you about the molecular and cellular underpinnings of human brain lateralization. So I have no disclosures, and all the results I am going to show you today, you can find them on the preprint posted on bioRxiv, and you can have it by scanning this QR code. So just to get everyone on the same page to start, hemispheric specialization is defined as the interdependence between a set of brain structures in one hemisphere and a given cognitive function. So in humans, for example, hemispheric specialization allows optimal language performances, and it's a fundamental principle through the entire animal kingdom, actually, and it's even more, and it's a hallmark of human brain organization. So usually when you think about hemispheric specialization, you are going to think about these two complementary functions that are language and visuospatial attention. So you can find in like 85% of people, the language processing dominant in the left hemisphere, as you can see on the image in blue there. And you will find visuospatial attention processing dominant in the right hemisphere, as you can see in pink in the image. And even if hemispheric specialization is that important for human evolution and cognition, actually there is a lot of things we don't know about. And more specifically, we don't know a lot of things about the molecular and cellular substrate of this lateralization. So the main hypothesis of this presentation is going to be that functional asymmetries emerge from distinct neurotransmitter, mitochondrial, and cellular profiles. And to do that, I'm going to combine fMRI data, PET data, transcriptomic from the recent paper from Zhang and colleague, and postmortem data from the recent paper of Mosharov. And I'm going to focus, obviously, on two main systems, on the language system and the visuospatial attention system. And to do all of that, I'm going to compute the asymmetry map for every of these data sets using the Aisha Atlas, that is a functional atlas specifically dedicated to study hemispheric specialization. So all this brain is just to show you what I'm trying to link together. So there is 4 functional tasks, 3 language tasks, a production, a listening, and a reading task, and 1 visuospatial attention task. And I'm trying to link these asymmetries with these 4 molecular biomarkers that you can see on the bottom right, and 19 neurotransmitter density maps, and 24 cellular biomarkers. And to do that, the first step I will take is to try to link these 19 lateralized PET maps across 9 neurotransmitter systems and the 4 lateralized functional tasks, so the language and the attention task. So to do that, I'm going to do a canonical correlation analysis, and the CCA is going to reveal a significant covariation between functional lateralization and neurotransmitter asymmetry. As you can see on the graph, on the x-axis you have the task mode, and the y-axis you have the neurotransmitter mode. And just to make things simpler, basically on the blue square you are going to find rightward asymmetry for attention that are going to be linked to rightward asymmetry in norepinephrine and leftward asymmetry in acetylcholine. And obviously on the other square, the red square, you are going to find the leftward asymmetry for language, which are going to be linked to an opposite pattern, so a leftward asymmetry for norepinephrine and a rightward asymmetry for acetylcholine. And what I wanted to do next is trying to link this acetylcholine norepinephrine axis that I identified with some molecular phenotype, molecular maps, so more specifically with mitochondrial phenotypes. So to do that, I perform multiple linear regression, and this regression shows that leftward mitochondrial asymmetries actually predict acetylcholine dominance. So actually that means indirectly it's predicting the left lateralized language activation, as you can see on the graph. So this result suggests that hemispheric specialization is supported by asymmetric energy demands at the cellular level. But furthermore, the third thing that I did is trying to link, once again, this acetylcholine norepinephrine axis with transcriptomic maps, so with 24 cell types. So to do that, I performed just a classic stepwise linear regression, and I identified 4 key cell types that are going to align with our neurochemical axis. And more specifically, I identify L5, L6, L2, 3 interneuron cells, which are glutaminergic neurons, and also microglial cells, which are non-neuronal cells, And notably, L5 and L2-3 neurons are going to play a key role in long-range integration, while microglia modulate circuit development and immune response. And finally, the last step that I wanted to look at is to test whether these molecular and cellular features actually can differentiate canonical brain networks. So to do that, I just perform a cluster analysis on the 7 classical large-scale canonical network that you can see on the top right here. So it's 7 usual networks: the somatomotor network, the frontoparietal, the dorsal attention, limbic, default, visual, and ventral attention. So I actually computed the average asymmetry value for the 7 markers that I previously identified, so the 3 mitochondrial markers and the 4 cell types that are all linked significantly with the neurochemical axis that I first identified. So once I characterized every of these 7 large-scale networks, I performed the cluster analysis. And with the cluster analysis, we can identify 2 main clusters, the blue one and the pink one on the figure. And the blue one is going to be mainly related to attention processing, while the pink one is going to be related to language-related processing. And which is interesting is that this big cluster, so this big pink cluster, is going to show leftward asymmetries regarding mitochondria and L5 interneuron, but also microglia. And obviously, or kind of in a funny way, it's the same thing with the blue cluster. They are going to show the opposite pattern, so rightward asymmetry for mitochondria and L5 interneuron and microglia. So we can find this double dissociation at the network level, and this opposing fingerprint is going to reinforce the idea that functional specialization is actually not just an fMRI thing or whatever, but that functional specialization is actually rooted in biological asymmetry. And I've been a little bit fast, but in summary, and to talk about some summary of the finding and the implication, we can say that lateralized cognition is actually underpinned by a neurochemical gradient, so this acetylcholine norepinephrine axis that I first identified, but also by mitochondrial asymmetries, so by asymmetry in the energy demand in the brain, but also by cell type distributions. And this biological gradient organized into 2 opposing hemispheric systems, a first system that is linked to language processing and which is leftward dominant, and another system that is rightward dominant related to visuospatial attention processing. So all these results are a step toward biologically constrained models of lateralization, and which would be really interesting to do in the future is to actually go at the individual level to look at inter-individual variability, because until now, all these results are actually based on populational maps, so there is no way to study any kind of variability, and even worse, there is no way to look at atypical lateralization, so people that basically have, to be sampled, a reverse brain organization, so a right hemisphere dominant for language, for example. So with the data we have right now, this open data set, it's impossible to look at things like that, so it will be a really cool thing to look in the future. And obviously it will be very interesting to look at the developmental and causal mechanisms that are going to lead to these cellular and molecular asymmetries. And I wanted to thank my lab at Rutgers University, so the Holmes lab, and also Siddhant Chopra that is working at Origin here in Australia. And if you have any question, you can ask them now, but you can also contact me on my email address or on Blue Sky, or you can try to catch me at the poster session in, I think it's poster 8-10. So thank you, everyone.
So there are no questions in the app. Does anybody have a question for the microphone? Could you come up quickly in the, interest of time? Right. If I may ask a question, or do you have a question? The axis, acetylcholine, norepinephrine. Does the data set, does Justine's data set, I should know, but I've forgotten, does Justine's data set have a acetylcholine transporter marker? Because what you saw was M1 receptor for acetylcholine and the norepinephrine transporter. So you're looking at neurotransmission there at two different levels. One is, the binding of the transmitter to the receptor, and the other one is the compound that removes the neurotransmitter from the extracellular space. I was wondering if so those are the only two that you found that were significant, right?
Yeah, but it's like a little bit more complicated. To make things simpler, I actually keep only like the two main neurotransmitter of the mode, but actually it's more like a combination of like the 19 neurotransmitters.
Right. Okay. So in the interest of time, you only
I sampled
okay.
It, but
a
bit more
time keeping, very good.
But thank you.
If I might just ask one further
There is a question.
Stepwise regression allows the first step to soak up most of the variance, and subsequent steps are dealing with the residuals. But that gives you a skewed view of how important the variable is based upon order of entry. So have you considered something like dominance analysis or relative weights analysis to get the actual proportion of variance from each of these contributors?
No, it would be a good idea, I think, maybe to look at that. For what I did like a, I don't know you call that in English, like a double stepwise regression. So it's like adding variables and removing them like at the same time. So I like trying to combine this variance problem. But yeah, I can also try a dominant analysis.
We have time for one quick yes/no answer. Did you look at other networks in the brain, or did you concentrate on the language and attention networks?
So no, I actually, to make things simpler also, I concentrate on the language and attention, but actually if you include other tasks like motor tasks or even emotional tasks, the main things that are like popping up are like language and attention, like these two, polar asymmetry are actually the ones that are like.
Right, so whoever asked this question, go and visit Loïc at his poster and you can hear all the details about the other networks, okay? And now we unfortunately have to move on to the next talk.
Thank you.